from yesterday set up online already on our Google Classroom. So you can just go to that and check that out. The other thing is for Friday, we'd like the students to prepare a one slide, Google slide about yourself that includes just a little bit about who you are, your interests, your school, where you go to, and any other fun facts about yourself. It would only require about 1 to 2 minutes total time of presenting, just so we all have a sense of who everybody is in our collaboration, because we are going to be working with each other throughout the next year and possibly the next couple of years. So I'm going to turn over the presentation to Mrs. Mackey. I'm going to stop sharing and we can start up.
Okay, so I'm going to take over and making sure I pull up the right one. Is that right? No, I don't like it. Can I just share my screen? Hello, anybody?
Yep, you could share the tab.
View tab.
I think what you were trying to do was share a tab.
How's that?
Yeah. Now hit slideshow. There you go.
There we go. All right.
Perfect.
Okay. So, so what we're going to be talking about now is the process of growing protein crystals. And basically, in situ, which is inside the cells, proteins are liquid and they're moving and they're constantly in just different forms and things. And the pure protein gets isolated as a liquid from a bunch of different organisms, but for our purposes, almost always from some sort of bacteria. It can be from a whole bunch of things, including humans and chickens. The work is done for us. Getting that pure protein is done for us by SSGCID, the Seattle Structural Genomics Center for Infectious Disease, which is why all of our proteins do play a role in some form of infectious disease. And they send them to us, and under the right conditions, we can work to get those liquid proteins to slowly come together and form a crystal with a set lattice, a set structure that makes it much more easy to visualize exactly what the protein looks like. Mr. Boland talked about yesterday how you could look at a single protein from 4 different angles, 10 different angles, and every different angle will be different as long as you're looking at the same structure from different angles. It's great. What you don't want is different structures, different versions of that one thing that you're trying to get because it was moving and there's something in a different place. You want them to be kind of stuck in place in this lattice. Sometimes the conditions that it takes to get these proteins to do this crystal formation are well known, and sometimes they're not. Sometimes scientists have to play around with what it's going to take to coax this protein into this formation. We prefer to the conditions as a conditions could include temperature, which can be room temperature almost all the time. Ours are, but they can be other ones. But then there's also this solution called a precipitant or a mother liquor that is mixed with the protein and used to help coax that protein into forming that crystal structure that we want. And there are in just an unlimited number of different precipitants and mother liquors that can be used. So the good news for this group is that SSGCID has provided us with a group of proteins with known crystallization conditions. In the past, and I'm not going to spend a lot of time talking about the past, but in the past, we would get the proteins, and part of your job as the student would be to research possible conditions. And then we were starting from scratch and it had less success than we might have wanted. So this precipitant usually has 3 components, some type of a salt, which is obviously not just table salt. It can be a whole bunch of different salt solutions, some kind of a buffer that makes sure the pH stays stable, and then some other kind of polymer that helps slow down the process of forming the crystals, so that as the protein forms into a crystal, it forms a nice lattice structure at the molecular level. So an example of one of these mother liquors could be something like this that I have here, where the ammonium sulfate is the salt and the 1 molar MES keeps things constant at a pH of 6.5. And then the 1,4-dioxane is used as that stabilizer we were talking about, the precipitant. So then what you do with this condition, this mother liquor, this solution that you have, is you add it into a reservoir of a well inside something called a plate. And I'm going to show you these in a second. And a plate has rows and columns of wells, so it looks like a grid. And each well has an area called the reservoir, which is where the bulk of this solution that you have identified is going to go. And then a smaller area where you mix that solution with a little bit of your protein. And this is what allows the crystallization to happen. So a plate looks something like this, and you'll get a closer look at one of them in a little bit, but you've got letters going down one way and numbers going across the other. So you could identify this one as B3, this well right here, and that helps you keep track of what's going on in your plates. Each well could have totally different conditions, or you might change conditions based on columns or based on rows. And this way you could duplicate the same conditions in 4 or 5 different wells and increase your chances of getting crystals. You could change the precipitant. You could change the ratios. So you've got those 3 different components to a precipitant. You could always change how much salt, the percent of the precipitant, stuff like that. The concentrations could be different. And then you add it to the well area. The protein and the precipitant, like I said before, are then mixed in a fixed ratio, which can also be changed. So you could do a 2-to-1 precipitant to protein, or a 1-to-2 precipitant to protein, or a 3-to-1, or a 1-to-1. All of these things are possible. And then the reservoir, basically the mother liquor in that reservoir starts to pull liquid water vapor from that drop as it starts to reach osmotic equilibrium. So close up, here's kind of what it's going to look like as that water leaves. That's what's slowly going to coax that protein into that crystal form. So this is a view right here of one well, one of those wells in the plate. And it's been filled, the reservoir here has been filled with the solution. Here they're calling it the cocktail solution, but I've called it the precipitant or the mother liquor. Then we have the drop of protein mixed with the mother liquor here. And this is showing you that you've got your cocktail solution and you've got your protein, and the initial drop are mixed together. And what happens is, as that water, as osmotic pressure takes over and water slowly leaves that drop, you should start to see these crystals forming in your drop. That's the hope anyway. And you're looking at it from the top, so you'll be seeing this drop, and inside that drop, you should start to see visible crystals. There are 2 different types of plates I'm going to mention because I don't know what your particular school is going to be working with when you practice with this. This is the hanging drop that you just met in the previous slide, where you put a drop on a cover slip and then you flip it over really fast, and then the drop is just hanging there. Or there's this other setup called a sitting drop, where there's a small little shelf where the drop of protein and mother liquor sits, and then the well is here. And it doesn't matter, the osmotic pressure does the same thing anyway. Some of them have the little shelf sitting in the middle. There are advantages and disadvantages to both of these types of plates. A hanging drop is harder to plate because you got to get the technique of flipping over that cover slip down. But once you have crystals, it's really easy to get the crystals out of there, because that's the next step. Once you have these protein crystals, now you have to get them and prepare them to be analyzed with an X-ray. So harvesting those crystals is a major step, and it's easier to do from a hanging drop plate. Sitting drop plates are much easier to create the plate, but then kind of a lot of people find harder to harvest the crystals. So once you have your plate set up, well, first the question is which one is better, and how long does it take until the crystals form? And that is a question that varies from protein to protein, from condition to condition, all sorts of things. A lot of the frustration we had in the past was that the answers to these things is, well, growing protein crystals is more of an art. And it's a frustrating art if you don't know what these conditions are, if you don't know the best way to grow your particular protein. And if it was a novel protein that's never really been studied before or deposited before, you're starting from scratch. Once you've done this, though, you have to screen your plate. Another way of saying that sometimes is we'll use the word score a plate. So you put the plate underneath a microscope and check one well at a time to see crystals, because sometimes they're visible with the naked eye, but most of the time they're not. So here's a picture of one well that has some crystals growing in it, very nice looking crystals too, and then some blobs of whatever. And then it's really important. You've got this plate with multiple wells, rows and columns, and every one of them could have something different in it. So it's really important to keep careful records. What is in each and every one of those wells? And then what kind of crystals did you get, if any? So here's an example of one of my students' data sheets when they were looking at one of their plates. This is the notes that they took as they're sitting with the microscope and figuring out what has worked for growing crystals and what hasn't. So like I said, keeping accurate records is super important. Here's a bigger plate. Bigger plate basically means smaller wells. The plates are all basically the same size, and the wells can just keep getting smaller and smaller. And here you can see we had some complete fails and a whole lot of other stuff that looks really weird. But the accurate records that you need to keep are not just the conditions. There's metadata, which is important for every scientific experiment to know, well, who did this work and who are they working for? What are they working with? The date that the plate was first created is important. The volumes of everything that you use, the ratios of everything that you used, and then each well's content. So this is all what we call metadata about each and every one of these wells, and it's important to keep track of it. You will be practicing doing all of this in the future with a known protein called lysozyme. I know we mentioned it yesterday. We're going to be talking more about it in the future. But this is to give you a feel for the entire process, for what it takes to make a protein plate and score it and go through the process. But the really good news for you guys as a cohort of Spark students is we have gotten through one of our Beamline scientists, we've gotten hooked up with Dr. Scott Lovell at Kansas University. And he is a protein scientist. And he has a machine that can plate teeny tiny amounts. One plate has 96 different wells. Or 96 wells. And so he can put 96 different conditions in every one of those things. And as it turns out, this is now how most scientists do their crystal growing. It is less and less common to be doing this by hand. That's kind of old school now, is what we have learned. So Dr. Lovell is fantastic for us. And what he does when he gets our proteins from that Seattle company, he puts a plate together. And then he shares his work with us through a program called RockMaker. And RockMaker has cameras on it at all times so that you can watch the crystals grow here from wherever you are. You can watch what they're doing in Kansas. So here's what RockMaker, one screen of RockMaker looks like. So this is called a screen. There's a lot going on here. This is one 96 well plate. It contains, and there's 2 drops in every one of these wells. So this one is actually set for drop number 1, which is the protein we've identified as S11. Drop number 2 would be S12. BRK refers to the known screening of chemicals. So when you set up a plate under BRK's conditions, that means that it's known exactly what mother liquor precipitant recipe is in every single one of these wells. And they're all different by just a little tiny bit. Rockmaker is also really nice because it has 2 different computers analyzing your drop to tell you whether or not there's a potential for crystals there. So everywhere where you see 2 red squares, it's probable that you don't really have crystals, but it's always a good idea to eyeball it and look anyway. And anywhere there's a green square, it tells you that the computer thinks that might be a crystal. Look at that a little closer. 2 green squares is both programs seem to think that there's crystals there. So this is, I've got kind of highlighted here, the well A6. So here is A6 close up, and it gives you the score that the computers have determined whether or not there are crystals there. And you can look at them and see just how pretty those crystals are. Down here, you could click on a camera. You could scroll backwards through time and watch the crystals form. And they're updated every few days, which is fabulous. So this is an example of what the Rockmaker interface looks like. And from what Seattle has sent us most recently at the beginning of the summer, and then we sent to Kansas, we have seven different proteins that are crystallizing under multiple conditions. So there's the one from A6 that we just saw. I think that was actually an S12. I think that's drop 2. And then there's this, and this gives you an idea of how different proteins can look. These are crystals. These are definitely crystals. I wouldn't call these good-looking crystals, but it's a start. Maybe we need to tweak that mother liquor a little bit in order to get these to be a little bit more on the symmetrical and organized looking side. These clearly are very, very tiny. Maybe they need more time to grow. Maybe they need their precipitant tweaked a little bit. Here, the crystals are kind of forming up here and there. That's something we'd have to look into. And these, I don't have the close-up of this, but each one of these little circles is clusters of teeny tiny needle crystals. So also probably not our best candidate for harvesting from this, but it's a starting point to take a look at what conditions this protein did this in, and then see if we can change it a little bit to get something that looks more like what we really want to look for. So each of these 7 proteins that we have grows under multiple conditions, and Rockmaker keeps track of every single one of them for us. So it's taking care of that metadata. So if we go back to that really pretty looking one, you just click on one of those blue tabs at the beginning, and it tells you the buffer that was used, all of the standard ingredients. It gives you the protein. This is the protein ID. Everything you need is taken care of in Rockmaker. So I'm assuming that later on in this, if not in this training session, once school starts up, you will be getting a little bit of exposure to Rockmaker. It's actually fairly intuitive to work. So down there is the ingredients. So our plan, our rough plan right now, because is to work with 3 of these proteins. It is a lot, and we can take some of the other ones and put them on hold for the future. We have 6 schools, and what we're hoping to do is to have 2 schools per protein. This way you can collaborate, but you'll each be working with different conditions since each protein grows under different conditions. So you'll be talking about the same protein and analyzing the samples coming from those different wells or different plates, because there's sometimes 2, sometimes 4 plates per protein, each with a different standardized screening kit. So once this happens, once your school gets their protein, you're going to need to do some background research. An essential step in all of this looking for proteins and finding out their structures is great, but what does it all mean? And so one of the most important things we need to do is do that background research. One of our best tools for this is something called the Protein Data Bank. And you're going to be learning more about this later on the week. But this is basically a history of proteins that have had their structures figured out. And so you'll be looking into whether or not it's already been resolved and under what conditions. Are there different variants? Are there different, has it been resolved but to a kind of weak resolution and we can do a better one? Have there been ligands with it or not? Are there related proteins to the one that you're looking at? That'll help give you insight into what it does. And then the scientific journals, which is where most background research also has to happen. This is what's known about the protein, what's known about what role does this protein play in disease? Why should we even bother studying it? And what organism was it isolated from? And what does that organism use the protein for? So all of these things are coming up down the road. And I just wanted to give you the heads up for that. But I have basically just kind of encapsulated kind of quickly how we get crystals to grow. And right now, how Dr. Lovell gets those crystals to grow for us. So does anybody have any questions? I'm going to take that as a no. I will be posting this presentation to the stream in the classroom. And if you're interested, because I went too fast or it was too much or you took a mental break, there's embedded in this presentation is a animated little video about how to grow crystals, but it basically just says everything I just said. I have.
All right. Thank you.
Okay.
Excellent. I love the pictures of the crystals on the Rockmaker. And just so the students know, the students are going to have access to Rockmaker as well. So you'll be able to go on. We'll give you the password. It's nothing crazy. And you'll be able to look at the crystals. And there's actually a way to score them. So we actually have the capacity to score the crystals ourselves. And then the folks over in Kansas can look at those scores and determine whether or not those are good crystals or bad crystals, because sometimes we think something is good and it's like, nah, it's really not anything that's worthwhile. The other thing is, as Mrs. Mackey had said, some of the crystals that kind of grew a little funky and look a little skinny, they can adjust those for us. So they'll change the concentrations. But keep in mind, those drops are only a couple of nanoliters. Those are not gigantic drops. So we're dealing with really small microscale chemistry here, and we're dealing with extremely small amounts of protein volumes. But they will scoop those crystals out, freeze them, and send them to us. And then we can shoot those at the beamline. I just want to show one quick picture that while you were doing this that I found, so that everybody kind of gets the sense of what we're talking about here. This is what the plates look like. They're 96 well plates, and the yellow blob here represents the well that rather the reservoir where the conditions go in. So you can put 96 conditions in here, and then these two small dots represent where drop 1 and drop 2 would go. So drop 1 would be in the orange and drop 2 would be in the blue, and those would be where you would set up the protein conditions. So technically you can actually do 96 conditions for protein 1 and 96 conditions for protein 2. And so that's how they have this set up.
That would have been helpful.
And what, yeah, I was, as you were doing this, I was like, wait a minute. I said, oh, this is it. So serendipity is what happens, you know? So what's neat is this lets us see exactly how the proteins are set up. And it gives us a sense of just how small everything is. And they're able to scoop those proteins out of those two little orange and blue drops and put them in a puck and then send it to us so that we can shoot them at the beamline. But that's what those trays look like. And again, the Rockmaker has it scans over these. So these trays all sit in this, what Scott described it as a protein hotel. And every certain number of days, the scanners go over it and they take pictures. And then they have these two separate programs that have the algorithm that predicts whether or not those are looking like actual crystals. So it's pretty neat how this runs, and it's pretty amazing how many proteins they can set up and run through in just one shot. Something like that would take us months to go through, so it's pretty impressive what you're able to do. So collaboration, that's what it is. Scientists all collaborate with each other and they all help each other out. Yep. All right. Mike, are you ready to go? Mr. Pinto, you all set?
Good morning, everybody. Yep, I'm all good. I'm gonna try and share my screen, make sure that it works. Let's see. Tab.
Yep, we see it.
Okay, good. And I'm gonna have a slideshow. All right, and you guys have the full screen slideshow now?
Yes.
Okay, all good. All right, so the next step comes with why do we freeze the protein? So, Miss Mackey went over the process of growing the crystals, and that could take anywhere from hours up to months, depending on the conditions and the proteins that you're working with. The question is, all right, what's the next step? And so in order to preserve those crystals, you need to freeze them. All right. And so first thing I want to jump back to is a little bit of high school chemistry, reminding everybody that molecules are always in constant motion. So if you visualize, what a gas looks like, the molecules are moving around chaotically and up, down, left, and right. Liquid, they're held together a little bit closer, but they don't have a fixed location. When you have a solid, they're locked into a fixed location. But remember that with a solid, the molecules are still vibrating in place. All right, and so they are still moving. They're not still. Okay, so now the synchrotron will take a picture of those proteins. And if they're moving very quickly, you're not going to have good resolution with that image. So think about if you have, you're trying to take a picture of like a car moving very fast, or a person running, and a train going by. Those things, when it's moving very quickly, you're not going to get a good image, okay? Because you have the object moving and you're trying to take a snapshot of it, but if it's moving, you're not going to have a good picture of it. So the goal is to try and get these protein molecules to move as slowly as possible so we can get a good image of them. All right, so we go through a process called cryoprepping, and we use liquid nitrogen. I'm sure you've seen liquid nitrogen at, sometimes with like a science magic show, or, sometimes your science teacher might have it in the classroom. Liquid nitrogen is approximately negative 196 degrees Celsius, or negative 320 Fahrenheit. So very, very, very cold. Remember that absolute zero is negative 273 Celsius. So, we're working our way close towards absolute zero, where molecular motion would stop. We're down there pretty deep, negative 196 Celsius. And so that helps to slow the molecular motion down as much as possible of those protein crystals. Now, one thing about crystals, you have a crystal is a repeating geometric structure. So with the X-rays that are coming from the synchrotron, just remember that they are not passing through one individual protein molecule. They're passing through many, okay? And so you have the X-rays passing through the crystal. In order to get a consistent image of what that one individual protein molecule looks like, you need every single protein molecule in that crystal to be oriented in to have the same orientation. Okay, so it's important to preserve that crystal and have all the protein molecules locked together in the same orientation. So the molecules, they must all, as you said, be in the same orientation in order to get good data. Now, cryoprepping does this, and it does the following as well. It helps to lock that molecular orientation in the crystal, okay? So that every single protein molecule that's in that crystal is oriented the same way, so that when the X-rays travel through that crystal, and we're going to go over that in more detail down the road, but when the X-rays travel through that crystal, they're deflecting in the same way, no matter which protein molecule they're going to hit. So everywhere in that crystal, all the protein molecules are oriented in the same way. In addition, the process of the X-rays passing through the crystal, it doesn't happen instantaneously. The loop that holds the crystal rotates over the course of maybe 10 to 20 seconds or so in order to get an image or a snapshot of the protein from multiple different angles. And that helps us to get a good three-dimensional view of the crystal, of the protein, so we can see it from multiple different angles. Now, as that loop with the protein is rotating, the X-ray, it's very, very, very high energy, and so it can do some damage to the protein molecules. And so having it at a very, very, very cold temperature helps to minimize that damage that is done by the X-rays, so that you still have good protein molecules throughout the whole 10 to 20 seconds that the X-rays are passing through. And lastly, as we said, it helps to slow the molecular motion down to prevent the blurry image. So if the molecules are vibrating in place, but they're vibrating very quickly, you're going to have that blurry image that's not going to be a very good image of the protein, and you're going to have a lot of uncertainty in it. And so by cooling it and bringing it as cold as possible with the liquid nitrogen, it's helping to slow the molecular motion down. And by slowing that molecular motion down, you're reducing the uncertainty, so that you're minimizing the blurriness of the image. All right, so we cryoprep for three different reasons there. Now, once the crystals are grown, we catch them. There's some process called fishing in cryoloops. Okay, and so this image on the bottom right, we have some decent crystals there, and you don't necessarily want small crystals because you're not going to be able to pick those up with the loop, and you don't necessarily want the entire drop to be crystallized because then you can't pick out the individual crystals. So you want a decent sized crystal. You want to be able to see the the geometric pattern there, so that you have a clear crystal there. And then you have to fish them. You basically hook them with a little tiny little cryo loop, and that image in the middle there shows, you can see it almost looks like a little hairline there. That's a loop. And going by hand with a microscope, you can fish for that crystal, and you get it inside that loop. And then that cryo loop, this is showing a zoomed in view of the cryo loop. All right, those might not be the best crystals inside the cryo loop, but it shows a good close-up view of the cryo loop. Once you've caught the crystal, you fished for the crystal in the cryo loop, you get it out of the hanging drop, and they get stored in things called pucks. So this shows a puck here, and each one of these little holes in the puck, you can put in a different cryo loop. So you might have a sample where you can get 5 or 6 different crystals, and you fish for each one individually with a different cryo loop, and then you can store multiple samples in that puck. All right, so the puck is it's a metallic structure with a whole bunch of different holes in there that the cryo loops can then get stored in. And then that holes, that these show the cryo loops. So the cryo loops have a tiny little loop at the tip, but then this circular part is what goes into the puck, so that you can, they kind of get locked in place. All right, this shows a different type of puck here. These look a little bit more like the ones that we work with, and they're numbered, so which cryo loop came from which sample. You can have 2 or 3 different proteins in there, from different samples, depending on what you've actually fished for. And this entire puck then gets stored in the liquid nitrogen. And the liquid nitrogen, it cools the metal, it cools the protein, it cools everything down to that negative 196 Celsius in order to preserve that protein crystal as best as possible. All right, so it all gets stored in pucks, and you get individual pucks, and then those pucks, you can store those for long periods of time in liquid nitrogen. As long as it stays cool, the protein crystal will get preserved. And then when you have your time to go to the beamline and run it, you get a chance to then load the puck, and then a robotic arm comes in and takes individual cryo loops out one at a time to then analyze the samples. And we'll go over that process down the road with the robotic arms and everything. So that's why we freeze the protein crystals, in order to prevent the blurry image. All right, slow molecular motion down, in order to lock the crystal in place. So having that cool temperature helps to lock the crystal together so that every molecule has the same orientation. And then also to minimize the damage of the radiation from the X-rays, because they're such high energy, and it takes a couple seconds in order to run the sample. So the cool temperature helps to prevent that radiation damage over the course of that process. All right, so that's why we freeze protein crystals. And again, I'll post that as well in the Google Classroom.
I
Right,
just have a
great.
question.
Go ahead.
Yeah, I'm sorry, I'm not really a biology person. So is the theory that the structure of proteins as you described it yesterday, with all of the hydrogen bonding and whatever else is holding it together into its structure, that really is pretty constant? And all we're doing now is crystallizing that structure into a crystal so that we can analyze it, but the structure itself doesn't change as we go through this process? Or can the structure be altered by this process?
So that's a good question. So the protein structure can get changed by a few different things. Like if you change a pH, for example, the protein structure itself can change. Those intermolecular forces can kind of change how the protein folds. So I do know that the proteins can change, and that's why it's important to note the conditions when you carry out the crystallization, because you might have, one posting in the protein data bank under certain conditions, and you have one shape of a protein, and then you have different conditions and a slightly different shape depending on how it folds differently. So it could be temperature differences, pH differences, different things like that can alter the shape a little bit.
Is there any effort, and I guess it would depend on how it crystallizes, but is there any effort to match the in situ conditions in terms of pH? I mean, I know certain proteins or enzymes respond differently under different pHs. Do they try to match that, or is it really just a function of how it best crystallizes?
So I think somebody else can answer this a little bit better. But my understanding is just that, it would be ideal to match that, but that might not be the best condition to actually crystallize it. So you would want to know what the structure is, inside the cell, but that might not be the best way to crystallize it in the dishes that we're working with.
Thank
It
you.
also has to do with the salt. Oh, go ahead, Sharon.
No, go ahead.
It also has to do with the salts that you have in the conditions too, because these conditions have different types of salt combinations. There are different kinds of buffers that they put in. So that can also affect it as well. And then the other thing that affects it is how the crystal grows. Some crystals will grow very ordered and very nice. So they'll be kind of set up in perfect rows, almost like desks in a classroom are set up. And then sometimes a few of the crystals might be set off at a little bit of a different angle because it's not as well ordered because of the conditions it was grown in. Sometimes they grow too quick. Sometimes they grow too slow. But most of the time, if they grow too quick, you get these really thin crystals that aren't really well ordered. And that can affect the results that you get because your x-rays are going through different portions of the individual molecules, and so you're not getting the perfect picture of what it looks like. So you can have that happen as well. And then basically you need to go back and try to tweak the conditions of it so you can get the better organized, better ordered crystal as well. So there's a lot that can affect it. And basically what scientists try to do is get the best resolution they can get. So that's why sometimes somebody will publish a structure to the PDB at a certain resolution, and then another group will come in and work on it and try some different conditions and get a much better resolution because they've gotten the crystals to be a little bit more ordered and they get a better resolution on it. And they get a better structure too, overall.
So I was just going to chime in and say
Yep.
the very unsatisfying answer is it depends entirely on the crystal. And as Mr. Boland mentioned, that yes, salts really can play a huge factor in how the proteins crystallize. Excuse me. Additionally, proteins like transmembrane proteins, very, very hard to crystallize outside of the membrane because they change their structure once you remove them from the membrane. So that can be very difficult. One thing that scientists will often do is try to compare what they've crystallized with predictive models. So if they can show that these predictive models match what they are getting out of the crystallization process, that's another layer to indicate that that's truly the structure of the protein in vivo. And then they'll start binding things like ligands and whatnot, and you can predict sometimes structure or potentially function with DNA sequence. So all of these things contribute to whether or not they believe the protein has been crystallized successfully.
Thank you.
And I just want to, because you know me, I have visuals all over the place. I have to. This is an image of what the crystals look like when they're ordered versus when they're not ordered. And you see how they're all kind of sitting at all these weird angles to each other. So when the x-rays come in, they're coming in at a specific angle. And when they diffract, you can kind of see these orange arrows uniformly. But if they're disordered, your diffraction now is spread out all over the place. And so you don't get the best look at it. And that actually happened with us with, I think, one of the beta-lactamase crystals, where it was just disordered and it wasn't sitting the way it should. And you can tell because your diffraction came out. We got some areas of the protein that looked really great, and then other areas that didn't look good. And when Kevin analyzed the output, he said, "Oh yeah, you can tell from the way the diffraction pattern is that this is not a well-ordered crystal." Because normally you want your crystals to look like this, where these are the individual molecules inside of the unit cell of the crystal, and they're all nice and ordered with each other. And so when the X-rays hit them, they're aligned really nicely. And these are different types of unit cells that we'll see. Like there's a C2, there's an I2, there's all sorts of different patterns that you get with that. And so this shows how, nicely they can stack and pack. And if they're not ordered well, that's when we get weird diffraction, and then we have to kind of go back to the drawing board and try different conditions. Okay. And I think that's it. Kelly or Sharon, anything else to add, or Jocelyn, or any other questions? Okay, so just a quick reminder for students. I know we have a couple of students that are on that maybe weren't here yesterday. Just want to remind you about the introductory slide. It's a short little slide that, let me just put this up here. Here you go. That will have you all present on Friday. So on Friday, we're going to ask students to introduce themselves. And again, be creative. You can put images in there, a minute or two max, just who you are, where do you go to school, what are your interests, what's your favorite subject or topic, and what do you aspire to do, and maybe some unique talents that you have. And this can all go in the introductory slide. If you look on our crystallization Google Classroom, you'll see it's the very first material that's posted in the 2024 week 1. It's titled Teacher and Student Introductions. And so students, you'll have access to that, and you can just add your own slides to that throughout the week. And that's it. All right, so thanks everyone. It's 10:45. So we always said we're going to be about 45 minutes to an hour. So today we're 45 minutes. So get outside, enjoy the great fresh air and the sun.
Seriously.
Yes, and we will see everybody tomorrow, 10 a.m. Have a
Bye,
great
Rob.
day, everyone.
Rob, just
Okay.
to let you know, I popped my slide into the presentation.
Oh, excellent. Okay, perfect. So you can do that Friday. We'll do that, or tomorrow. Okay. Have a great day, everyone. You can all log off.
Take care, guys.
All right.
Bye, guys.
All right.